Hello everyone. Today I'll be taking another Viva Live session with QQSphere on the subject of microbiology with the topic immunology and vaccines and the subtopic type of immunities. So let's start.
Welcome to the examination. Let's begin by discussing the basic differences between innate and adaptive immunity. Can you outline the key features of innate immunity and how it responds to pathogens?
Yes, innate immunity is the first line defense. It is present from birth. It is fast, non-specific and lacks memory. Components include skin, mucous membranes and phagocytes.
That's correct. Now, within innate immunity, how do phagocytes recognize and engulf pathogens? What kind of receptors do they utilize for this?
Uh, I don't know. Can you explain, please? Can you explain?
Phagocytes use pattern recognition receptors or My apologies, I will clarify. Phagocytes use pattern recognition receptors or PRRs to recognize structures called pathogen associated molecular patterns or PAMPs on the surface of pathogens. An example of a PAMP is lipopolysaccharide. Are you familiar with any other types of PRRs?
Um, Other types of PRRs are TLRs, toll-like receptors, nod-like receptors, RIG-I-like receptors, and C-type lectins.
Excellent. Now, moving on to adaptive immunity. Can you explain the concept of immunological memory and how it contributes to a more robust secondary immune response?
Uh, Adaptive immunity is also called acquired immunity. It is a highly specific defense system that identifies and destroys particular pathogens, providing long lasting memory driven protection. It adapts to new threats by utilizing lymphocytes, B and T cells to generate antibodies, eliminate infections, and establish immunological memory for faster responses to future reinfections.
Yes, that's right. Now, what is the specific role of T helper cells in coordinating this adaptive response? How do they assist B cells and cytotoxic T lymphocytes?
Helper T cells are crucial white blood cells produced in the thymus. They coordinate adaptive immune responses. They detect pathogens and activate other immune cells such as B cells to produce antibodies and cytotoxic T cells to kill infected cells.
Precisely. Let's now talk about types of vaccines. What's the fundamental difference between live attenuated vaccines and killed vaccines, particularly regarding their ability to induce long term memory?
Vaccines are classified by the technology used to train the immune system to recognize pathogens. They include live attenuated, inactivated, subunit or conjugate, mRNA, toxoid, and viral vector types. They work by stimulating infections with harmless components, stimulating immune uh, memory without causing disease.
You've listed several types of vaccines, which is good. However, can you focus on the distinction between live attenuated and kill vaccines regarding the strength and duration of the immune memory response? Which generally provides broader, longer lasting protection and why?
Yes, live attenuated vaccines use a weakened form of the germ to create a strong, lasting immune response. They require fewer doses. An inactivated or killed version of the germ cells are vaccines which create immunity, which cannot cause the disease. These are often used for pathogens that cannot be weakened and typically require multiple booster shots.
Precisely. Now consider a clinical scenario. Suppose a patient presents with a severe allergic reaction immediately after receiving a vaccine dose. What type of hypersensitivity reaction do you suspect? And which immunoglobulin class is primarily involved?
The immunoglobulin class primarily involved is IgE. and it's a type 1 hypersensitivity reaction.
Excellent. Finally, can you briefly explain the principle of herd immunity and how it protects vulnerable populations who cannot be vaccinated? What threshold is typically needed?